Yeah, hello. Yeah. Yeah. Hello, everyone, and welcome to this another afternoon of an essay public lecture here. I hope I'm audible. Yeah. Welcome again, everyone. And here we have today a public lecture by a very prominent person in his field, Professor, who will be Nazar, who will be giving us a talk about it. I request Professor plus Director TFR and Professor Ankana to please grace the stage. As I was saying, Professor Nitzan is preeminent professor at University of Pennsylvania in chemistry, and he's also a donor professor of physical science and professor of chemistry. I wish Professor Rafi.
Thanks, Parag. So welcome to the hybrid ASET in the Department of Chemical Sciences public lecture by Professor Ibrahim Neetsan. I'd like to first call Professor Jairam Chengulud, Director TIFI, to just say a few words to get the proceedings going.
Yeah, thank you, Ravi. I don't have a great deal to say. Welcome again on behalf of TIFR to this public talk. This talk, as we will hear shortly, has been the funding for this talk and as well for the visit has been provided by the Sarojini Damodaran Foundation, which is basically operated by Mr. Shibu Lal. And this funding is actually very critical for us, I should say, because we do like to take part in a lot of these public outreach activities. We'd like to have eminent people visit TIFR. It enriches us, it enriches the academic environment of this city. But these are things which are sometimes a little difficult to do with government funding. And this is where private funding, I think, really has a very important role to play. So we're very grateful to Mr. Shibulal as well as the Sarojini Damodaran Foundation for funding this entire event and making it possible. I'd also like to thank Professor Nitzan for very kindly accepting our invitation. He's been here for a week interacting with the department. He's given several talks, of which I think this will be the last on this visit. I'm certainly looking forward to this talk. And I do hope, Professor Nitzan, that we will have the opportunity to have us visit us, to have you visit us again. I won't give a detailed introduction to the speaker. We'll hear about that shortly. I'll just end again by saying welcome, and we all look forward to the talk.
Okay. Thanks, Jairam. Now I'd like to call Professor Ankuna Datta, co-chair, Department of the Chemical Sciences, to tell us a few things about the Sarojini Damodaran Fellowship.
Okay, welcome to the Sarojini Damodaran Foundation lecture, public lecture. So before Ravi introduces the speaker, let me tell you a little bit about the Sarojini Damodaran Foundation and its links to TIFR. You'll be seeing many more of these lectures in the coming months. So the Sarojini Damodaran Foundation, which actually funds this visit by ABE, was established in 1999 by Shri S.T. Shibudal, who is the co-founder of Infosys and the former CEO also of Infosys, and Kumari Shibudal. And this was done to transform the lives of underprivileged in the society. The formal philanthropic journey of the Shibudal family actually started in 1998 with the establishment of a scholarship scheme in S.T. Shibudal's hometown in Kerala. So now this foundation is under the umbrella of the Shibulal Family Philanthropic Initiatives, and the activities bring in sustainable improvements into the lives of underprivileged through education, healthcare, organic farming, and social welfare. Now, SPFI or SFPI's activities, which is what the foundation is, is the acronym is, has scaled through years and has impacted the lives of several of thousands of families across India, and of course, also our TIFR family through the Sarojini Damodaran Fellowship, which is named after Mr. Shibulal's parents, Sarojini and Damodaran. And this initiative also started in 1999 at the very initiation of this foundation, which shows the commitment of this foundation towards science and research. So the key goal of this program has been public dissemination of the excitements of science and technology and bringing in eminent researchers who can interact closely with researchers in India, and also allowing students to travel to different labs around the world. And I'm sure many of the Sarojini Damodaran student fellows are now in the audience who have actually traveled using the fellowship. Also, the visitor here, Ibn Itzan, with whom we have had an excellent interaction, very close interactions throughout the week. So the fellowship has definitely contributed to the growth of the scientific community at TIFR in Mumbai and Bangalore also at large through the support of these public lectures. And so we at TIFR thank the SFPI for being a part of our scientific journeys and for being a part of, bringing us together with researchers like Nitsan. Thank you.
Thank you, Ankuna. Okay, so we are almost ready to hear Abe speak, but before that, I just want to give you a brief bio sketch about Abe. So Professor Abraham Nitzan is the Donner Professor of Physical Science at the University of Pennsylvania. So Professor Nitzan received his PhD degree from Tel Aviv University in 1972. He then carried out postdoctoral research at MIT and the University of Chicago before returning back to Tel Aviv University as a faculty in 1975. He became a full professor of chemistry in 1982 and then stayed there for over 3.5 decades, I think close to 3.5 decades. In 2015, he joined the University of Pennsylvania as professor of chemistry, and in 2018, he became the Donner Professor of Physical Science. Professor Nitzan's research focuses on the interaction of light with molecular systems, chemical reactions in solids, liquids, or interfaces, and also the energy and charge flow in these frameworks. He has published over 350 papers on these topics, on this topic, and he's one of the most highly cited theoretical chemists in the industry. He has received many awards and recognitions, such as the Israel Chemical Society Prize and Medal, the Emmett Prize, the American Physical Society's Plyler Prize, and the American Chemical Society's Theoretical Chemistry Prize. He is a member of many prestigious scientific societies, such as the US National Academy of Sciences, the American Academy of Arts and Sciences, the American Physical Society, and the Israel Academy of Arts and Sciences. In 2010, he received an honorary doctorate from the University of Constance in Germany. Now, finally, while closing, I'd just like to note that Abe is a committed educator, and his book, Chemical Dynamics in the Condensed Phase, is one of the most widely used. Is widely used as a graduate level textbook in the field. Okay, so with this, I'm sure we're eager to hear Abe talk. So before that, I just want Professor Chengalur to just felicitate Professor Abe Nitzan, and then we can go forward to it. Okay, so Abe, it's all yours.
One, two, three. Do you hear me? Okay. So, good afternoon. I want to start by thanking the Institute and the family for making my visit so nice and pleasant. Also, my host, Ravi. I was here for a week and I talked to many people and I really had a wonderful time. So I said I look forward to visit you again. This was really a very great experience for me. Now, Ravi told me only, say, I think about 3 weeks ago, so that asked me to give a public lecture. And public lectures are the most difficult one to prepare because usually I come, I saw some equations, everybody get impressed, and that's about it. But you do not do this in public lectures. So I went to my hard disk to see what I gave in public lectures before, and it turned out that this was all for high school students, which is not exactly the audience here. But I still will show you just a few slides that I usually show to the student. This is how I start. This is basically how chemistry starts. And very soon people ask what it is good for. And this was when they tried to get gold out of the things. And hundreds of years later, this is still the issue. This is a poster used to lure students to study chemistry under the promise that you can really make good life being a chemist. But this is from 100 years ago, so you can ask whether this is still true for chemistry. And I found the answer last week in the New York Times. It looks like this. You really can make money in chemistry even today, it appears. So let's see, also becoming a bit more serious, this is where chemistry as a science really started. This is the first book ever published on chemistry by Robert Boyle. You see the word alchemist here, if you can read it. Here, and this hypostatical is, this is where you really ask serious questions about chemistry at the time. And what I want to do today is tell you what is serious about chemistry today, because chemistry had hundreds of years to develop, and it is really a very mature science. But in the past 50 years, it was really went into transformation where people started to look at chemical processes on the almost individual molecular scale. So, when we compare a molecule to a football, it's just like comparing a football to Earth. So we really went down to individual molecules to look how they react, how they convert one form of energy to another. Chemistry basically, if you want, you can say chemistry became physics, because when you look at processes on the molecular scale, basically you look at the physical processes that underline chemistry. And just to demonstrate it, I will show you a reaction that I used to play with when actually when I was a child. Does it move? Well, the child, there was a store that was irresponsible enough to sell me sodium metal, and I was going out in the rain when there was puddles of water, and I, with my friends, threw these pieces of sodium into the water, and this is what happens when you do that. It's a really very vigorous reaction. Eventually, it ends with an explosion. We can move it faster ahead, so we do not have. Actually, it's going to the end. So this is what happens when you react sodium metal with water. But if you take a single water molecule and a single sodium atom, and you bring them together, nothing happens. You see the difference between macroscopic chemistry and microscopic chemistry. You need some more than one molecule and one atom to make this reaction happen. And the reason is that the energetics is very different. You need solvation, you need to form hydrogen molecules. When you react sodium with one water molecule, you do not have enough hydrogen atoms to form an hydrogen molecule. So you see that going microscopic will makes a difference. So I want to tell you what we learned when we investigated chemistry and chemical processes on the microscopic scale. But the way I want to do it is to also introduce the basic notions that serve us to understand when we try to understand what we observe in this phenomenon. Timescales is one of them. That is, timing is, of course, very important in many processes. You do not have to go to the molecular level to understand that timing is important, but relative timescale is another important thing. If this dog tries to get to this hamburger and he has to cross a bridge which will break under his weight, the dog can still make it to the hamburger if it will run fast enough. So it will not stay enough time on the bridge. It takes time for the bridge to get destroyed. And if the dog runs fast relative to this time, it will make it. This is clearer even here. See, the bird has to run fast enough on water in order to stay on top. When we realize that relative timescales are important, we can go back to chemistry to see what are the important timescales in chemical process, and they span many, many orders of magnitude. The fastest chemical processes occur on the, I don't know if you can see the numbers here, but these are femtoseconds, 10⁻⁻15 seconds. We can go on the scales. There are chemical processes that take place on seconds and minutes, and different processes. When we compare, when we try to analyze what makes processes go, the relative timescales of the different phenomena that take place become extremely important, and I will show you how this plays when we do our study. The other things are length scales. There are different length scales involved in many phenomena in nature. Molecules reside in the nanometer range, which is here. But you compare different sizes of things, and I will show you examples where the length scales become critical to understand how the chemistry evolves in time. Here is one example. The material in these tubes is the same. These are nanodots, small particles made of this composition. The only difference between them is the size of the nanodots, and the size controls the color, because what determines the color is the relative size of the dot and the wavelength of light. When you change this relative size, you get very different colors. You see how size plays in this phenomenon. Another concept is symmetry. This process I'm showing you here, in fact, is one of the hot subjects under research these days in chemical systems. What people observe is a strange phenomenon where what you have here underneath is a piece of metal. This is gold in this case, and you use light to photoemit electrons from the gold. And it turns out that when the electron goes through these helical molecules, this is DNA in this experiment, what comes out are electrons with their spin aligned in a particular direction, in this case, pointing towards the metal. Now, this is something that nobody guessed can happen in chemistry. Spin is this property of electrons that control magnetisms. If all the electrons are pointing in one direction of the spin, this is where we have magnets. Some materials are naturally magnets because this is what their electrons are doing. Gold is not one of them. Gold is not a magnet. You emit electrons. These electrons come evenly, spin down and spin up with the same ratio. But after they go through these molecules, the ratio it becomes uneven, down spins dominate, and this basically is a magnetic phase now. And this is still under investigation, but there are strong evidence that the symmetry of this molecule, this chiral symmetry that you see here, is the dominant factor that makes it. But this is all the subject under current research. You know that birds navigate when they fly between continents. They navigate by sensing the magnetic. Questions will be great. No, this particular experiment I'm showing here, you come with light, you photoemit through a layer of molecules. What you have on the other side is a detector. There's a card called Mott polarimeter that can detect the spin of the electron and tell you whether this is spin down and spin up. This is the experiment. In this process, there's just emission of electrons and they go to the detector. There are other experiments in this field where you have two metals. Electrons come from one metal, they go to the other metal, and you detect them in the other metal with magnetic fields. So this will be more closely to what you have in mind. But I brought this here now as an example where the symmetry of the system appears to be the dominant, the controlling factor that makes this happen. And I mentioned this is just as a speculation at this stage. We know that birds find their way by sensing the magnetic field of Earth, but nobody really knows how do they detect the magnetic field? What is the process in their biophysics that makes it? One speculation is that this effect I showed you before, that molecules can control the direction of spin, is at the base of this process, how birds detect the direction of the field. But also the length scale becomes important there. I said that this helical symmetry is what makes the electron align its spin, as I showed you before. But this is also helical symmetry. But an electron is so much smaller than this structure that I don't think electron will care whether this structure is helix or anything else. The relative size of things again plays a role. The size of the electron and the size of the helical structure have to match in some way for the process to occur. So this is how relative length scales again come into play. Now in quantum, in these molecular processes, quantum phenomena becomes important. Quantum interference is an important subject. What you see here is what classical bullets will do. Right? There are two slits. You see two peaks there in the target. But if this was electrons with wave phenomena, we'll see interference pattern. So quantum interference become an important factor, and I will show you examples how this plays out. Tunneling is another important quantum phenomenon that starts to be important when we look at processes on the molecular scale. And if we could really control it and make it into a useful transport, this probably could solve the traffic jams I saw this week in the streets of Mumbai. Let's see, and cooperativity. I use this picture. This is not from chemistry, but from biology. These are bacteria that you put in some food and substrate that contains the food. They grow, but they grow in these particular patterns. My friend, this is Eshel Ben Yaakov, he died a few years ago, but when he took this photo, this was under research that it came under, the title he gave it was a bacterial intelligence. Somehow bacteria know how to grow to form these beautiful structures. I personally would not know that I would call this intelligence, because we see this also in, now let's see if we can make this move. How do we start this? Okay, great. So what I'm about to show you, this is a chemical reaction. There are no bacteria here. They just mix two components together and chemical reaction takes place. But in this particular reaction, this is what you see when you observe the reaction in a Petri dish. So you have this plane, and as the reaction propagates, you get these wonderful structures. We had some music before. I don't know why it is so, but okay, you see the principle. The music is less important here. Anyway, eventually you're going to see really patterns that you could call it molecular intelligence, but I would rather not, because this is really results of how physics behave when things become nonlinear enough to create such structures. So bacteria seems to do the same thing, not necessarily because they are intelligent, but because physics tells them to grow in this particular way. Here is the music. Too late. Okay, let's go on. So these are the concepts I want you to keep in mind when I tell you more about what we do in this nanochemical phenomena that I'm going to tell you about, and I will try to do it without equations, this being a general audience. So this is the last transparency with equations I'm going to show you today. If I forgot to erase equations, just disregard it from what comes next. So the one thing to, let me go back to the time scale things. I said many phenomena are controlled by relative timescales between different processes. And this is not special to nanochemistry. Even when we do things on the macroscopic scale, we see evidence of this importance of relative timescales. This is one of the simple chemical reactions. Chemical reactions, many of them are just barrier crossing phenomena. I got from one type of system to another through a barrier. And in order to go through the barrier, I have to have enough energy to cross. And the simplest way to calculate the rate of chemical reaction is to assume that actually it's all done in equilibrium. If you assume that there is equilibrium in this region, then you know what is the probability for molecule to be on top, because this is what Boltzmann tells you about potential energy, e to the minus energy divided by kT, T is the temperature. And you know what is the probability to have velocity. So you know that how many molecules are here and what is their velocity. So you just compute how many molecules go out. And this is called transition state theory. And this is just equilibrium. Time scales do not play any role in this argument. But this is really just an approximation, because you assume that there is thermal equilibrium here, but if molecules escape, there will be less molecules on top than in chemical equilibrium, so you will overestimate the rate of reaction. And you assume that all molecules that go out will really remain out, but some of them will collide with the environment and go back. So not everything that appears to go out really go out. So also overestimate how many molecules go out. So the rate that you calculate is an upper bound to the actual rate. It is always larger, it is not exact. If you want to be exact, you have to look at the relative timescales. What do I mean by this? There is the molecule trying to cross the barrier. It has to get energy from the environment. It has to basically, these are collisions with the molecules of the solvent. If there is no collisions, the rate will go to zero because there is no way to get energy in order to cross the barrier. So without collisions, there is no rate. If you have too many collisions, basically the parameter in the solvent you look at is the viscosity that tells you how much I collide, how much I fill the solvent. When it's all become very viscous, I just cannot go out. So again, the rate of reaction will go to zero. So really, chemical reaction rates as a function of the coupling, the strength of the coupling between the molecule and the environment behave like this. They go to zero when the coupling, friction or viscosity, same thing, when the coupling becomes very small, also when the coupling becomes very large. Transition state theory is always above. And the good news is that most chemical reactions are in this regime, that the properties of regular solvents put the friction that the molecule feels in this regime. So the transition state theory we use is a good approximation, but it's always an upper bound. But the relative timescales are what determine this shape of the dependence of chemical rates on properties of solvent. Let's go to another class of reactions, electron transfer reactions. They are, again, extremely important class of reactions, appear in photosynthesis, in vision, in corrosion, in batteries. And the rate of chemical reactions is controlled. I gave a lecture to the students earlier this week. It is controlled by the relative timescales of the electron trying to move and the environment fluctuating around this, around the molecule that undergoes this electron transfer. How do I mean? Electrons go very fast. When the electron makes a transition, electron is so fast that on these timescales, the environment is static. It does not move. Amount is much slower than the motion of an electron. So electron goes under a static environment, which means that during its flight, it cannot take energy from the environment, and energy has to be conserved, so electron can go between initial state and final state. And when the fluctuation brings these states accidentally together, this is where the electron jumps. This is how the relative timescales play out. And when we try to around the molecules, and they arrange themselves according to where the charge is, but I cannot of the dipoles here and here are different. And to go, as I said, the environment is static during the transition. I cannot go this way. I have to get these fluctuations of the environment to give me into situations where the two electronic states on the right and on the left have the same energy. Then I have electron transfer. In a diagram, it looks, let's leave this. In a diagram, it looks like this. What are plotted here are two potential surfaces. They describe the electronic energy as a function of the environmental configuration. One of them belongs to the case where the electron is on the donor. The other is the state where the electron is the acceptor. The minimum of the surface is the stable state. And these two minima are at different energies. Electron don't jump between one minimum to another minimum because this is the nuclear configuration axis. Nuclear configuration cannot change during the jump of the electron. Electron can jump only when the two energies. This is energy as a function of nuclear configuration. Electron can jump only when the nuclear configuration is such that the two energies are the same. It is this point where electron transfer takes place. Can you say it again? There is no other point where the energy is the same. Energy, if I have two curves describing the energy as configuration, only when they cross, the point where they cross is the point where the energy is the same. Right? This is the energy axis.
Yeah.
And this is the configuration. And there is only one point where at the same configuration, the energy is the same. This is this point. Okay? Which means that if I start here, I cannot just go down. I have to go through this point before I can come down on the other side. In other words, what controls the chemical reaction is not the energy of this relative to energy of this. Usually, you know, if you think I'm here and I'm going down, it will be more efficient. But really what controls the electron transfer is the fact that I have to reach this point. Now, whether you followed me or not in these details, you can appreciate the following figure, which shows you again this figure and just the message that for the electron to go from this state to this state, he has to climb to this point. And now if I look at the rate of electron transfer as a function of how much downhill I go, here I go more downhill, and here I go even more downhill. But in every place, I have to get to this point. You see that the activation energy is positive, it becomes 0, and then it is positive again. So you will predict that as a function of how much downhill I go, the electron transfer rate will be faster and then slower again. This is how it looks experimentally. In these different molecules, they took a series of molecules where, as you go to the right, the molecules on the right, the reaction is more downhill. But the rate first go up and then goes down, and then goes down. This experiment was done in 1984. It was predicted by Marcus in 1955, and 6 years after, or 8 years after the experiment was made, Marcus got the Nobel Prize for this idea. This is basically all the idea. The rest is mathematics to show what is the actual rate based on this idea. But just this diagram is the essence of what Marcus theory predicts, that the activation energy does not depend simply on how much downhill you go in the reaction. So these are all macroscopic phenomena, but really what I want you, what happens on the single molecule level or a few molecule level when we go to nanochemistry. So at some point in time, like 30 years ago, 40 years ago, people asked, now that we understand electron transfer, molecules, you can say molecules conduct electricity. It can go from one side of the molecule, it can go to the other side. Can I use a molecule as an element in electrical circuit? And maybe if I'm more ambitious, can I make a transistor out of it? That is, transistor is made of 3 electrodes, a source, a drain, and a gate. A gate is used to control the state of the medium between the 2 electrodes. And if this medium is a single molecule, this can make a really tiny transistors. And this would open a field of molecular electronics. Actually, molecular electronics has been open and is a very active field. And people are constructing such transistors, they work. They are not technologically yet useful as devices because of a phenomenon I will show you in a moment, but certainly useful to make interesting headlines. And this is what you see in the laboratory. I want to show you, to emphasize the difference between macroscopic conductor, Ohm's law. Ohm's law tells us that the current as a function of voltage is this red line, linear line. Current is proportional to voltage. The slope, which is the conduction, is constant. And what is plotted here is what you observe for the current as a function of voltage, certainly not a straight line. The derivative, which is the conduction, is certainly not a constant. This is the derivative. But another thing you observe is that there are several experiments here. Each line is a particular experiment, and every experiment gives you somewhat different result. There is noise. This is why you do not yet make, unless we learn how to control this noise, we cannot make useful devices. I don't want my cell phone to give me different signal every time I open it, right? Here is one reason why you can get noise in this system. You do the experiment again and again and again. You see different results every time, but when you plot them, you get every line here is different experiments. And in these particular experiments, they all fell on 5 or 6 lines. And when you collect all this data, these are the data that you collect for all of them. If you divide them by integer, you can put all of them on the same line. It turns out what you see here is some junctions. There are a single molecule, sometimes there are 2 molecules, sometimes there are 3. And every time you form a junction, it gets be formed with different number of molecules between the 2 electrodes. This is a source of irreproducibility, but we understand where it can come from. There are several other reasons why noise appear in these systems, and once we nail how to control it, we may in the future perhaps have useful devices made out of them. Another thing in these devices, you know, in Ohm's law, if I make the voltage larger, I get larger current. I make the voltage smaller, I get smaller current. But in molecular devices, the 2 time scales, you remember, the electrons go fast, the environment, the nuclear motion of the environment is slow. And every time you have 2 time scales, you can have, if I send the voltage up, electrons move fast, but the environment still did not move. When I take it back, I get a different path because the environment is not the same. We get the phenomenon called hysteresis. We see a lot of hysteresis phenomena. You see here these examples. When I go up and down, the current as the function of voltage behave differently. It is not a nuisance. This is actually one source when you can make useful devices using this hysteresis phenomenon, but I will not have time to go into details there. This is, I remember I mentioned interference. When electrons move, they can interfere if there is more than one pathway. If electrons could go this way and that way, the 2 pathways can interfere. They can interfere constructively or destructively. Like in this junction here, there are 2 ways for the electron to go between these 2 electrodes, either through the bond below or the bonds above, and they can interfere constructively or destructively, depending on where I tie the electrode to the molecule. I can make the contacts here, or I can make the contact in another configuration. And you see here constructive and destructive interference exactly because of this phenomenon. When you see here low current, this is destructive interference, high current is constructive interference. This you will not find in macroscopic physics. Another thing you can do with these junctions is put lights on and try to control them with light. And if I can call a single molecule, we can control the state of a single molecule with light. There is an example. With light, we can open or close a bond. And when we do this, we can change the conduction of the molecule. So we can make a switch made of one molecule. And here is actually how it looks in an experimental device. This is the 2 electrodes, and this is a molecule between them. And in this device, a UV light will switch the conduction from low to high, and then visible light will switch the conduction in the other direction, and you have a switch based on a single molecule. Again, not something that you can do, that you can do in macroscopic physics. But in these junctions, not only electrons go from one side to the other. Every time you pass current through a conductor, you also generate heat. To understand the phenomena of the performance of such devices, you also have to study heat conduction through a single molecule. So you can plot cartoons like this, but in fact, you can make an experiment that looks like this. Many of you probably recognize this as an STM scanning tunneling microscope picture. You come with a sharp metal tip. You can connect a single molecule between a metal tip and a metal electrode. Usually the signal that we observe is conduction. Electrons go. This is what scanning tunneling microscope is doing for you. But people use similar devices to also study the heat conduction through a single molecule. And you get result, you can actually measure it. In these 2 lines, one of them is electron conduction, the other is heat conduction as a function of the length of these molecules. You can calculate these things and get results that are pretty close to experiments. So we have control both experimentally and theoretically on understanding heat conduction through a single molecule. And one interesting phenomenon that we observed recently is that you do not have interference also only in electronic conduction, what I told you before, but you can also have interference in heat conduction. because phonons, again, if I connect the two electrodes, this is called the meta configuration to these two atoms, or to these two atoms. This is the ortho configuration. This is the meta configuration when I connect in there, or the para configuration. I get different heat conduction just because of the different ways I connect the hot and cold walls to this system, because again, the heat or the phonons can have two passes to go, and they interfere on the other sides, and they can interfere constructively, and I get high heat conduction, or destructively, and I get low heat conduction, even in this in heat conduction. In fact, this paper really had trouble going into literature. It was published after 6 reviewers went through it, because the first reviewer just rejected it, saying this cannot be true because you did a calculation using Newton equation, classical, and everybody knows interference is a quantum phenomenon. But interference is not a quantum phenomenon. Interference is a wave phenomenon. And classical waves also interfere, and phonons are waves that are heat waves going through. So this, you can get it quantum mechanically. In fact, these are the classical results, and these are the quantum results. They are not the same. The quantum result here and the classical result here are the same. Quantum and classical are different here, but you still see the effect of interference, both quantum mechanically and classically. So another factor, interference and tunneling, but many times it is much easier to look at classical mechanics because we have intuition for classical mechanics. We ourselves live in a classical world mostly, but many things that we understand from classical mechanics play also in.
So this is a theoretical calculation, right? Or
there is no experiment yet. This is
difficult to do.
This is a prediction. People measure now heat conduction, but nobody measures heat conduction in a system that has two distinct pathways for the heat to conduct. So this is a prediction. Yeah. The interference of electrical current has been seen experimentally now many times. Another concept that I mentioned is cooperativity, and this is the last thing I will tell you about today. Remember, I showed you this bacterial growth and these interesting patterns that chemical reactions may make, because molecules sense each other. This was in macroscopic physics, but this is a phenomenon that we observe in microscopic physics when in very, very small systems. What you have here is basically two mirrors. This is a metal plate, this is a metal plate. Light, if I put light on this system, it can be reflected between two mirrors and get trapped between them. And when light gets trapped between two mirrors, it changes its properties. It is like a particle in a box. Particle in a box can be in any energy. Free particle can take any energy, but particle in a box have discrete quantum states. And the light is the same. When light is trapped in a cavity between two mirrors, it can occupy only particular wavelengths, which mean only particular frequencies. And when we put molecules between the two mirrors, they interact with these cavity modes. This is what we call these distinct frequencies that light can exist between two mirrors. The molecules interact, and that's the interesting phenomenon. I will show you the experimental evidence. They do not interact separately, each molecule by the light. They interact as a collective. They interact together with the light. I will show you how we see it experimentally. The light have one particular state. That is, there can be no light. And if there is light, there is one particular state, as we said. And the molecule can have a ground state and an excited state. And the excited state of the cavity, when there is a photon in the cavity, state of the molecule, if they have the same frequency, they interact with each other and they create a combined and hybrid entity. We use the name polariton for this hybrid entity. It's a mixture of light state and matter state. It is not light and matter separately. It is a mixture of both of them together, this polariton. And we see the signature of these polaritons is these two peaks. If there is no molecule in the cavity, we see just one peak, which is the molecular peak, which is the cavity mode. This is a discrete peak because we said the cavity can support only particular states. And when we add molecules to the cavity, we get two peaks. These are the two polaritons that are formed. We call the molecular transition and the cavity transition. They interact with each other. When we increase the density of molecules in the cavity, these lines separate. When you do the theory, you learn that the separation between the two peaks measure the strength of the interaction between the light and matter inside the cavity. And actually, we can quantify it. The strength of interaction depends on the number of molecules. I don't know if you can see that there is 1/2 here. It is the splitting, this distance, as a function of the square root of the number of molecules, square root of the concentration. The coupling increased like the square root of the number of particles. This is the coupling per molecule. The coupling per molecule is just a number, but the coupling you observe experimentally is not the coupling that a single molecule feels, it's the coupling that the collective of molecules feel. And this coupling can become to be, here the numbers are relatively small. This is 120 wave numbers, is about 1/20 of molecular energies. But in other systems, this can be of the order of molecular energies. So you can ask the following question. If the coupling that the molecules feel with the cavity mode is so large, can it affect chemistry? That is, can I observe different chemistry if I put the molecule in a cavity? That is, if I just put the molecule between two mirrors. Now, you do not expect if you put, well, there are some legends about not putting yourself in front of a mirror or between two mirrors, which can do something to you. But this is real chemistry. So here is a question. We know that when I put many molecules together in a cavity, they interact collectively with light, and this interaction can be very large if I have enough molecules, because it's the number of molecules that contains it, that determines this. So if I have enough molecules and have strong coupling, can chemistry be affected? And if this is true, this will be dramatic, because usually we do not think of a chemical reaction, So before molecule crossing a barrier, we do not think of this as a collective phenomenon. If I have many molecules, Avogadro number of molecules, I do not expect them to collectively cross the barrier at the same time. It is a stochastic process. Sometimes this makes the reaction, sometimes on the average, we see a reaction, but we do not think of this as all the molecules together, like soldier marching, going from product to reactant. So is there really a collective chemistry going on in the cavity? Now, the experimentalists find that there is. These are experimental results done by a group in Strasbourg. And what is plotted here is the ratio between the rate of chemical reaction. Details are here, but this is a chemical reaction. And this is the rate, the ratio of the rate inside the cavity when I put the reactants between two mirrors and outside the cavity. And this is plotted as a function of the same coupling strength that you saw before. These things that become stronger when the number of molecules increase. So really what you change is the number of molecules, and you see that the rate, this is the rate per molecule. Usual chemical rates, they depend on the concentration, but no, a chemical rate is some coefficient times the concentration or product of concentrations. I'm talking about the coefficient. The coefficient is a constant, does not depend on the number of molecules. But here it does. So experiment tells you this amazing phenomenon that the molecules, when you put them in the cavity, they somehow do collective chemistry. So you can imagine when these results came out, they started to come out 10 years ago, 2015, every theoretician that did interaction of light and matter stopped what they are doing and rushed to try to understand what is going on here. And we are such people, so we rush to understand what is going on here. So we did calculations. There are other experimental results. Let's not spend time on that. Our calculations. What you can put molecules in between two mirrors on the computer and do the calculation. And we calculated all sorts of equilibrium properties. They are named pair correlation functions, time correlation functions, equilibrium distance between atoms, everything in equilibrium. And these are our results. Every graph here is two lines, one in the cavity and one out of the cavity, and we do not see any cavity effect at all. And they are not alone. There are these theorists all over the world. They're trying to look for ways to explain the experimental results. There is way ahead, right? This is an equilibrium calculation, so maybe the phenomenon that dominates in this experiment are non-equilibrium phenomena. This is a semi-classical calculation. Maybe you have to go to full quantum mechanics. People tried it. It does not work. It is not that there is no effect of the cavity, but only when the cavity is so small that the coupling becomes very, very large. One thing I did not tell you about the coupling, it does not depend also only on the number of molecules, it also depends on the volume of the cavity. And the smaller the volume, the stronger the coupling. We will not go into details here. So you can get effects only when the cavity is so small that the coupling becomes very strong per molecule. But in these experiments that I showed you before, the cavity is very large. This could not explain what is observed. So we have a problem, and I'm not going to tell you what the answer to this question is because I do not know. I want to leave you with a question. But there is a future here. See, this is actually, we are now collaborating with the experimentalists. This is a grant we got recently from the European Union. This is the title of the proposal we enter, Unraveling the Mysteries of Vibrational Strong Coupling. This is the name that is given to the phenomenon I showed you before. There are this experimental group, there are two experimental groups, there are two theoretical groups, and hopefully 5 years from now, I will be able to come back and tell you what the answer is, but I cannot tell you now. And this is all I wanted to tell you about today, except thank you very much for being here.
Thank you very much, Professor Netizen, for a wonderful talk. And it's open for questions. I request students to please carry the mic to this.
Hi, Ib. I had this question maybe since I've heard the last few days. I'm sorry, a little bit again asking maybe it's a repetition, but have you looked at reactions inside the cavity which are slow but are dictated by flow of reactants going through? Because that will capture some bit of the non-equilibrium phenomena which you have not computed in these efforts.
So several people did. So we look at this is a chemical reaction, but you can forget about chemical reactions and ask about rate of flow. You can look at energy flow, and you can look at charge flow. And yes, you see effects, but in order to, how do you do experiment with energy flow? You have to excite somewhere and to see how the excitation go. So light is involved. So it is not only molecules in a cavity, is molecule plus light in a cavity. And the fact that cavity affects light, there is no question about it. I saw it before, it's like particle in a box. So to make a transport experiment that does not involve light becomes a little bit more involved. There are some you can do it with calculations. And there are some calculations for exciton motion that will predict that exciton will go faster. So light is not involved. You start with an excited molecule and you see how the excitation goes. But of course, excited molecule can generate light. So light is still involved. So it's one of the things that is in our proposal and need to be investigated more.
Thank you for the nice talk. So the question is like, the theoreticians are trying to reproduce the results only from the experiments, or they're trying any other reaction, new reactions?
No, we do not try to do a calculation on this particular experiment. When you look at a particular chemical reaction, there can be other effects that you're not familiar. You want to look at the generic model.
Yes.
So what is a molecule is energy is molecular energy levels and transition between them. So if this is really this kind of effect that the molecule interacts strongly with the cavity mode, it should be observed on any molecular model.
Yeah.
So we use simple molecules. So this is a very complex organic reaction. We look, all the calculations that we done were on system like water.
Okay.
Water or water with ions, things of this kind. So you can say correctly, you do not calculate this particular reaction, and therefore you do not know what happens in this particular reaction. But the effect depends on the particular reaction. It cannot be just a general effect of cavity mode interacting with the molecule. So this is something that, again, has to be clarified in the future.
Yeah, because the transient state, what we are studying, the vibration, what we are targeting, is there any relation between the transient state and the vibration being coupled? Or why we should choose a particular vibration?
Vibrational energy transfer is one of the things that came up. You remember when I told you that barrier crossing rate behaves like this, that it is going to zero when the coupling to the solvent is very, very small. It's going to zero when the coupling to the solvent is very, very large. So one possibility is that the cavity environment affects the coupling to solvent, the rate of energy and change. And if you are here, this will give you a cavity effect. So yes, this is an open thing that needs to be studied.
Yeah.
You're completely right.
Stating it for water. Has somebody done the experiments for water, and in water or whichever system you are seeing, they are still
People did show the polariton splitting, this splitting between two lines that depend on concentration. They did it in water, yes.
the experiment, and then your theory is predicting it should not depend on the concentration.
No. All we say is that we see the Rabi splitting, this splitting between two lines that increase with concentration within the calculation all the time. But we do not see effect on molecular properties. This is a property of the hybrid molecule light. If you look at properties of molecule, what do I mean by this? Correlation functions. If molecule move in one way in time t1, what is the probability it will do this at time t2? This kind of correlation functions. Or if I put an ion here, what is the probability to find an ion there? These things we do not see any effect of the cavity.
Have they measured these things and they're seeing effects on that or not?
Some people measure. There is a paper, for example, that tell you that they find that the dielectric response of water actually was different in and out of cavity. Dielectric response is also a correlation function of the type that we are calculating. we did do this calculation. We did not find an effect on the computer. So again, there is discrepancy between theory and experiment. Yeah.
So, very simple question. When you're going to cavities and mirrors and everything, you have surfaces. You're putting something between two surfaces, and there are surface effects. There are confinement effects. So, can one not explain such phenomena, for example, by invoking those effects rather than thinking about the hybrid modes?
So, you know, the world is large, and there are people who are very enthusiastic about it, and there are people who have doubts about it. And those that want to express doubts will form. Maybe you do not see the effect of interaction with the cavity mode. Maybe you see an effect of interaction with the surface. These concerns were raised. Usually when they were raised, the experimentalists who do this experiment came back with answers that look satisfactory, but then they are raised again. There is a paper by, there is this guy Simkin in the NRL. This is the one of the American national laboratories, and they did two experiments. One that came in support that is also showed the effect that Ebbetson sees, and this was sigh of relief by people like me. The effect is real because somebody else who really had doubts. I met Simkin a year earlier, and he said, I do not believe any of this, and now he reproduced such results. So this is very encouraging. Last month, he published a paper. The last sentence say, it is not every time we publish negative results, but I think it's about that. They do not say the experiment is wrong, but they just say there are some things in the experiment, not exactly surface effect, but some aspects of the experiment that will affect the experimental results, and they were not taken into account in previous analysis. So we have to look again. The field is in flux, it is evolving, and these things go on.
Thank you. If there are no further.
About the surface effects and the confinement, the surface effects have been taken care by having the even the cells or cavity, everything have the same interface. Even if I have gold, on top of the gold, they put some surface material like any polymer, which is kind of same in all the setups, like cell or cavity. The confinement effect also, you have off resonance, something called off resonance, where you have cavity modes, but they are not coupled. So they are kind of like control experiment.
I should add that with this size of cavities, also in the calculation, we do not see any confinement effect.
I think there is no more questions. I welcome everybody to.
Sorry, there is a question on Zoom. I don't think there's any question on Zoom.
No, I don't think there is.
Okay, so let's thank Abe for the wonderful talk.
Okay.
And please join us for high tea.
Thank you very